right, welcome to Reptile Fight Club. Sorry, it took me a minute to remember what podcast it was. It's been a little while, but I guess not that long. Welcome to Reptile Fight Club. Here we go for another fight, I suppose. With me, Justin Julander, is Rob Stone. How's it going?
It's great. Ahoy, hoy. Yeah. Longer since we've had an episode published than since we've done them. And maybe get a couple in here in short order to get back on track and all after the trip. So, yeah, all good.
Yeah, I saw Eric put one or two up. So, or the one that we did with them and then the one that we did together, right? I think I saw those come out.
So at least the one we did with them. I don't know about the one that we did subsequently.
Okay. I think there was something that got released. Anyway, when is what is when. So Great. Here we are now. Yeah.
Nevertheless.
Yeah. So yeah, things have been pretty crazy since the trip, and just trying to dig myself out of a lot of work and stuff at home, and we're getting ready for my daughter to come home from Texas, so she'll be home on the early December, so we're excited to have her back, and she's been gone for a year and a half, and so that'll be fun. But yeah, other than that, it's just business as usual. Snakes seem to be doing well. I've started pairing stuff up and getting stuff going, seen some locks in the womas and blackheads, and so hopefully we'll have an Aspidites year, get better than last year. Man, everything just kind of crashed last year. So see what happens. But yeah, how about you? You got stuff paired and going for the next season?
No, I don't really have anything that. Well, I suppose the Solomon Island tree bows maybe coming eventually. Although they're all kind of, at least those females are in heavy feed modes. So I'm not sure this will be a year. They kind of. I think we talked about this before, but basically it seems like they have lean years and heavy years where they're willing to feed or not willing to feed. Mm-hmm. And then in general, coming off of heavy years is the year that they seem likely to produce. So I don't think we're timing into one of those at this point. They're probably heading into a heavy year. Mm-hmm. So yeah, I'm not, with the stuff I have, I'm not pairing stuff up. Although the point of interest was not this past weekend, but the weekend before, we had power out for 25 hours when it was in the 20s outside or whatever. So that was, always the item of concern. And where I'm at, there really isn't, the question is always, you have a generator, and all this stuff. And it's like, I don't have a place that I could run a generator that it wouldn't be, that it would be secure, for sure, like with certainty, So I did get, at my wife's advisement, I wasn't even aware they existed, but they work off the 1 pounder propane camping, the sort of camping deals. Yeah. And they're approved design to be run in indoor conditions. You still need some ventilation, but man, those things definitely work, and at least takes the edge off for sure. Yeah. The funny bit is that with the stuff that I have, particularly the West Indian bow, is they had this, I strongly believe in light cycles. So it just turned into 25 hours without the light, kind of evening into the next evening. And the humorous bit was once the power kicked back on and warmed up for maybe 40 minutes or so, all the West Indian boas, the lights are still off because, the red bulb and all that. Yeah. And they were all just in hunting position, just like, it must have been a weird eclipse or something, but ready to, so
back at it. Yeah. Business as usual. Yeah. I'm sure wild animals need to deal with these kind of things all the time. I mean, they pop out of nowhere. A lot of, I think they've got to be adapted to have sudden drops in temperature. I always laugh, when we lower our temperature by a degree a day over the course of a week or two, for our cooling periods. It's like nature doesn't mess with that. It's just like one day it's 100, the next day it's 40, well, at least around here, maybe.
Right. I mean, that is the classic Colorado, Utah thing where you go, honestly, it must have been, frontier times, ignoring everything else. It must have been a super dangerous place to live in the sense of just how rapidly we can go from beautiful weather to terrible and vice versa. Mm-hmm. But, just everything looks fine. And the next thing you know, you got 18 inches of snow.
Yeah.
You know?
Yeah. And I don't know. I think we've softened as a people, like being out in Australia and having that heat, and just thinking, man, what, and frankly, I shouldn't have been trying to sleep when it was 90. I should have been out, scanning the rocks for geckos or something instead of trying to sleep, which was, just not productive. But I guess I was tired or something, but I wasn't thinking.
Yeah.
Should have been out shining the rocks, but yeah. So what do you do? But yeah, I think the times have softened us. Yeah, we upgraded to new phones and that whole nonsense, and it's just a headache and a half. And half the phones don't work, and like we're trying to get them connected to, the whole Apple thing. And oh god, my daughters are freaking out because they can't access their Snapchat or whatever they need to get on, and so it's been a little rough. We've become soft, I think.
Right.
Yeah. I'm like, just pretend like you're me when I was your age and I had no phone, but there wasn't even phones, Dad. You wouldn't even know what you're missing. Yeah. I'm like, well, we'll survive. I promise you. This is first world problems, so. Yeah,
absolutely. Well, I mean, it's just like, back in the 80s and 90s, right? You'd fix a nice supper and then you'd take a Polaroid picture of it and you'd walk around to your neighbors and show it to them. It's just the same.
Exactly.
That's exactly what we used to do.
Yep. Just like the good old days. Crazy stuff. But I mean, technology's nice. And this morning I was remarking to my wife how nice it was to take a hot shower on a cold day, how easy we have it and how comfortable we have it. And when those things go away, we realize it real quick, when there's a power outage.
Absolutely.
Or, when we moved into our old house, we found out that there were some things that were done incorrectly. The house wasn't up to code. And they turned off our gas because there was a carbon monoxide issue,
nice.
And that just about killed my wife. She was going to go take a nap, but then she decided not to, or, the carbon monoxide alarm was going off. And they called the cops and they said, yeah, if you would have taken a nap, you wouldn't be here today. So,
that's wild.
And then they shut off our gas until we got it fixed, brought up to code. And we're like, how did this get mixed in the, inspection that we paid for? Why did they not bring this up? And the realtor kept saying, well, welcome to home ownership. We're like, come on. Is there no recourse? Well, there is, but it's very expensive. And we're like, okay, say no more. We're poor. We get it. No justice for the poor people.
Right.
So we had to have a couple cold showers, and yeah, it's pretty, we're pretty soft these days, but anyway, enough of that. Things are good in the herp room. The babies have all started, for the most part, started feeding okay. I've got a couple holdouts, but Aunt Regia will do that. This year was fairly easy as it goes, with the ants. I didn't have the best of luck with the pygmies. I think being gone for a few weeks, that always kind of messes that up because they kind of need, at least once a week assist feeds. And so I lost a couple of those. It was kind of a small clutch anyway, and they were pretty small babies. So, but I have, a couple left, but hopefully next year I'll figure it out. That's what I always say.
Right?
Yeah.
Absolutely.
Yeah. Well, what else? Anything else we need to bring up? I think, I don't know. I think I'm just ready to fight. I just have this fight in me. I'm kind of worn out, actually. I started playing water polo, so we had practice today. I don't even know if I can play in the games or anything because I'm not a student. So it's like this 50-year-old guy is probably some weird guy coming to play water polo with the college kids. But I don't know. It's been fun. It keeps me in shape and hopefully keeps the pounds that I lost in Australia off. But so still can hit that second notch on my belt that I couldn't hit before the trip. So
very good.
Yeah, that 135 miles did me some good. I think we just sweated it all off with the heat.
Yeah. Right.
So yeah, my brain is not as functional as maybe it could be, but we'll go for this topic anyway. We had a topic suggested by a listener, Mark Heath. he was asking, should there be a fixed set of rules for taxonomy? And so I think, we've kind of hit on taxonomy here and there. I know we had Phil Wolf and was it Casey Cannon or who? Can't remember who Phil fought with about taxonomy. But anyway, right. Yeah, it might have been Casey. But anyway, so we've had a couple of shows on it, but I don't know that we've kind of hit that topic. And so I think there's some nuances to taxonomy that maybe is not that we could benefit from chatting about. And I'm trying to think of a good way to put the topic, but, I guess, pros and cons to fixed set of rules or a standard in taxonomy maybe would be a good way to state it, but I'm sure we'll branch off of that a bit and talk about the things. I mean, obviously we are not taxonomists. And so, we're just using kind of our educated, best understanding of taxonomy. And so we might be off on this. We probably ought to find some kind of taxonomist to have on at some point. But I mean, I've chatted with actual taxonomists and people who understand this much better than I do. And they seem as frustrated with some of the same things as I am. So talking with Rob before the show, we've got a few bones to pick with taxonomy, I guess. And so I think that's maybe a common frustration with people out there that, that taxonomy just sometimes drives you crazy because it seems so arbitrary at times. Other times it seems like, well, why would they make that choice? What is this, you look at the rationale and you just don't agree with it. So we'll kind of hit on these topics while we're talking about it. Thanks, Mark, for the suggestion. We appreciate it. And we appreciate you guys who are listening in and making suggestions and giving us ideas for topics or telling us what you want to hear. So let's keep those coming in and keep us on the right topic, I guess. So we don't get off on some weird tangent that we want to talk about. All right. So I guess let's go pros and cons maybe to a set rules of taxonomy idea. We'll go ahead and flip a coin.
Okay. Tails.
It is heads.
Heads. Okay. That's just as well.
Yeah.
Again.
I mean, I don't know which one I'm thinking about it, which one I prefer, but I'll go with, I'll go with pro. I'll go with pro.
Okay.
The taxonomy could use more a fixed set of rules and, ideals from group to group, but maybe I'll lead off. Why not?
Okay, cool.
I like to talk, just spout out my nonsense. So I think that's one of the big frustrations, right, with taxonomy that a lot of us have is that it just seems to be arbitrary. It seems to be dependent on which group of reptiles or mammals or whatever is being described. It seems like whoever's doing the research, whoever got the funding kind of calls the shots. They get to choose what model they use. They got to choose which DNA they think is important and which regions they're going to look at. And I guess to some extent, there are some definite challenges to having a set or fixed set of rules for this kind of thing. And I think they are kind of moving towards that in some way. But I do think, that it's necessary to have more than just one thing. I mean, I don't think you just get the DNA and say, okay, here's how it shakes out. We're just going to go with DNA. You also have to look at maybe physiology or how they interact with the environment, biogeographic barriers, things like that. And I think all those kind of things are taken into consideration these days. So, I do think there are some kind of overarching guidelines and rules to taxonomy. Maybe not as rigid as we'd like. I think our understanding of what taxonomy is, at least my understanding, is that it's not necessarily a firm science. I think you gather data and you interpret it the best you can. And it is up to the researcher to kind kind of identify what's important. And I don't know how we get beyond that, because the people who are interested in it and who are doing the work, who are putting forth the effort, why shouldn't they kind of give their best ideas forward? Now, some of the frustrating things are like, are subspecies a thing? It seems like a lot of people say, oh no, there's no such thing as a subspecies. And other people say, no, subspecies are a necessary thing. I think in the end, as long as we know what we're talking about and we can kind of see, and I think as I'm all scattered, aren't I? But anyway, I think as herpers, we kind of say, here's what's important to us. This is why we want these to be different, because they're different localities or different. So a lot of times it makes it easier if these different localities or these different types have a subspecies or some kind of name. Now, when
designation, right?
Exactly. Yeah. Scientific designation rather than just our trade names or our trade whatever. I think we like science to agree with what we see. And sometimes I'll say, herpers see these differences before the taxonomists do. I think for the longest time, people, that were into green tree pythons knew that the southern green trees and the northern green trees of New Guinea were very different. And they could tell, oh, this is a southern or this is a northern. And in fact, they are fairly divergent and different species. And so all these crosses between, Arubiacs and all those kind of things are actually hybrids of two separate and divergent species. So anyway, my rambling, I'll stop it and just kind of say, I think there are some general rules as much as can be done. Could there be more? Maybe. It's hard to say. Yeah.
Okay, fair enough. Well, as you say, there's a ton there. So let me take a couple of jumping off points, and then we'll just continue down the road. I think some of the variability that you've highlighted right at this point is based on there needs to be some variability amongst different group taxonomy of different groups because of variable rates of change within those groups, right? So they'll kind of their rates of divergence differ across these different organisms. So it can happen more quickly or more slowly depending on the sort of underlying genetic and physiological structure of those animals. Sure. In the same way that we've talked about with reptiles in general, particularly those coming from isolated populations, seem relatively immune from the effects of inbreeding in a way that isn't the case really with mammals, even in those small isolated pockets over time, right? Mm-hmm. So there's just sort of, they're not all the same, right? In the same way we've talked about with, why are there all these different goanna species within these different areas? If you can treat them all like, take the frank, they're all goannas, right? And the answer is, well, obviously they're not all the same, or they wouldn't have speciated. They wouldn't be different things if there wasn't a reason for them to be different things. Right. So I do think there's some element of truth to that, right? In terms of fundamentally, or at least fundamentally as a concept, I can accept that different groups speciate at different rates. And so there can be some variance in how we perceive different levels of difference across different groups of organisms over time.
And I would kind of understand that as like, say something that reproduces very rapidly, has lots of offspring, lots of chances for, genetic mutations or changes to occur in a short amount of time. Yeah. So they might change more rapidly, might be able to adapt more quickly to changing environments or situations over time. And so they might persist. Whereas something that takes 75 years to reach reproductive status.
The tuatara probably aren't. Yeah.
Exactly. There's a reason there's only a small isolated area where you can find tuataras in the world because, they don't adapt very quickly potentially. So that's, I guess kind of how I understand it, if that matches with your understanding as well.
Absolutely.
So you
probably expressed it a little better than I did. Yeah.
You might not apply the same rules to a tuatara's evolution as you would to a insect that
right, at least conceptually, right?
Yeah,
and maybe the answer is that you could apply the sort of same paradigmatic principles, but that when you're actually looking at it saying, okay, a lot of this, at least the way I understand it, is based on sort of rates of change over time and how long it's been. The perceived difference between most of this, right, is in the context of drawing distinctions amongst different things. It's less so than talking about a singular animal or type. It's how different is A from B, or A from the tree, right? And where do we wind up, and what's most closely related to the next closest item, and trying to make I think that's basically it for all of it, right? Is trying to make a tree that makes sense, and where we can wind up with some sort of disfavored naming or losing old names that have, been sort of we've all grown accustomed to or familiar with or like. That can be the result of, oh, well, this was described 1 year earlier, and actually where to make the tree make sense, then we're going to get rid of this name in deference to this other one or whatever, causing sort of radical shifts. I will say, just because it would be crazy to miss it, and I'm staring at the board here with all these colubrid and colubroid banners and all this different stuff, that Dr. Zach Loafman is actually a taxonomist, principally working in crayfish, right? We hear him every week talking about colubrids and colubroids, but he actually is. And so, I know their podcast. They've gone into the Eastern rat snake group. They did an incredible episode talking about kind of all the science associated with that. And actually that leads me right to a point that you had touched upon earlier, but that to me is kind of the fundamental underpinning of my whole understanding of this, is that taxonomy really, to me, is one of the softer hard sciences. So it is a hard science. I acknowledge that. And as a lawyer, I'm certainly not, sending strays here, right? Because certainly my stuff is as soft as it can be. But, fundamentally, I think we need to understand taxonomy not as a binary system, but rather as trying to take our understandings, progress them as far as we can, and then make the most compelling arguments that we can. And it's really not a function of what the author says so much as the receptivity of the audience to the persuasiveness of the argument of the author. Right. That's really what matters. People put out papers all the time, right? And some of them are taken up and others are not. There is a system of priority. And I think that's the stuff that when Phil was on, that he was really getting into, right? A sort of deference to the naming conventions and all those things. And that's really not kind of the focus of what we're talking about today. But to me, the key is, whenever over more than a decade of listening to Reptile podcasts, if I hear someone say, well, the science says, nothing great like that in the context of taxonomy, because in reality, the author of that paper, bringing their own personal biases to the party, is just trying to make a compelling case. And, I can buy or sell that, right? And ideally, when I'm doing that, I'm an informed consumer. And as you highlighted, as a keeper, particularly if it's something that I have a ton of exposure to, I actually might have more exposure to those as living creatures rather than just sort of DNA samples. Pickles or DNA samples than the people writing that paper. Like, certainly, that's absolutely the case in the context of the Ganyasoma paper, right? That always becomes a topic of consternation. Right. But that's the classic prototype example for me, because of my relationship with that on several different levels, that I can look at it and say the paper itself espouses that, well, based on the evidence that we saw, there are multiple different positions or alternatives for how we could lay this out. And a reasonable consumer of this paper could choose to think one of those multiplicity of options is the best, makes the most sense, fits their paradigm and their bias. But we're lumpers and this is what makes sense to us, so this is what we're doing. In the paper itself, if you actually read the paper, it says there are multiple things that could make sense here based on sort of what we bring to the party. We're choosing this option and we're freely owning that. The key point is that, to me, when it's then discussed on a podcast or whatever, it's just, oh, this paper in 2010 says it's this, so it's this. No, that it does. The paper itself doesn't even make that claim. They're making an argument. They're hoping to persuade. And ultimately, in scientific naming, two things. The first would be that really it's the uptake by the community, and that mostly means scientific community. We as herb keepers kind of are a little bit more loose with what we'll take or leave. But
Yeah.
in general, it means, they're putting an argument into the world. And the question is really, how many times is that cited further? How many later works accept what the argument that they've made and incorporate it into their own work, right? That's really what's important. That's the validation. So, yeah, absolutely. That's sort of fundamentally, I think we just need to get that out there because that's a bugaboo for me personally.
Yeah, no, I completely agree because, they're doing the best with what they have. And I think, you see that if you go back 10, 20, 50, 100 years, you're going to see a change, an evolution in the literature. You're going to see the first python species were all genus Python, they are all pythons. So they're all related. They're all in the same genus, and then over time, well, this one's a little different because it does this, or they've been apart from this one for millions and millions of years. So we'll put them here, and these guys moved in the Pleistocene. So we'll move them here. So, all these different changes and rearrangements and reassortments, and, people had just frankly the wrong ideas where they said, okay, these Aspidites have no pits. And so that must be the original Python form.
Those must be the most basal rather than the most derived. Yeah,
exactly. And then it turned out to be the opposite. They're the most derived. They're the newest changed pythons on the scene. And so, and DNA and other things have kind of helped us sort those things out. Now, DNA is not a magic bullet. You can't just say, oh, I did the DNA and now we have everything and we know everything about this animal. And I admit that kind of is hard to understand why. But when you think about the millions and millions of base pairs that go into DNA and trying to interpret what all that means, you can sequence the whole genome of a different animal or species and compare it with that of another. But what's important in that giant, huge mess of a DNA? And so I think as our technology rapidly changes and evolves, our methodology to analyze that also changes. And frankly, most of the regions, we have no clue what they code for, or what they mean, what they do. I think, the genome is still largely a mystery in a lot of things. And, for some things like the human genome or mouse genome or things, we have a lot better grasp and understanding on a lot of things there. Some, of course, like proteins and coding areas that code for proteins are probably well known, and we can look at that across, and it's probably fairly homologous with other, animals and so we can make comparisons and things. But I was just, I attended a lecture today on prion disease, and prion disease is a misfolded protein that can actually convert other normal proteins that are in our brains into the misfolded type. So it's like an infectious protein disease. It's very weird, but one difference in one amino acid change in that sequence of the prion protein results in a different kind, a resilience to that change in certain animals. So like they had to change the mice so they could get human prion disease because their prions were not the exact
That's totally wild
sequence.
Right?
So they're very similar. Yeah, they serve a very similar function, but the sequence is slightly different in those. And so that one change makes them resilient to, that kind of thing. So, lots of just needle in a haystack things that we're trying to figure out and trying to work out. And people spend their whole careers studying one protein and how, it misfolds and causes disease. And so, they know a lot about that one protein in that one way, but what does the normal prion do for, there's not a lot of knowledge on even what function it serves in the brain. So we of course there probably is some knowledge on that. I'm over exaggerating here, but we don't understand a lot about, a lot of things. The more you learn, you don't know much, and so a lot of it's kind of a mystery. So yeah, we can sequence it, but then how do we get meaningful information from that huge sequence dump? It's kind of
absolutely.
Yeah. So, and then how do we make that make sense in regards to taxonomy or change? It's not as simple as just saying, oh, I have the DNA sequence. Okay. Problem solved. Now we've got the DNA sequence, now we know. Is it helpful? Potentially, there may be things we do understand, like, oh, if we look at this, the mitochondrial DNA sequence, that can help us understand rapid changes because those are only inherited from the mother. And so you can draw certain conclusions and see certain changes over time. And so there are things that are known that are helpful, and we've been relying on those. But new advances come along and we say, oh, now we're looking at this and this is more important. And so that area evolves and changes over time too. So with our ability to get more information, our understanding of that information also changes and evolves. So, the papers of 10 years ago may look silly to us now because we went, oh, they didn't look at the genetic sequence. Ha ha ha, fools, And in the future, they're going to be looking at back on this time and saying, oh, they thought this. Oh, the fools. So it just changes. So, but back in the day, we can look at these old plates drawn in the 1800s and we can say, oh, that's a diamond python. I recognize that. I know what they're talking about when they describe its behavior or whatever. So, granted, some of that may be a little mixed up in history. There's an example in knobtail geckos, like an Underwoodosaurus and Nephurus. They drew a picture of one, but it was probably the other one that they named, and so that name should have gone to a different gecko, but that's just kind of how things shook out. And we don't need to go back and fix that because that would just confuse everybody, because we know what an Underwoodosaurus milli is and what a Nephurus levis is. And if you switch their names just because it took precedent because this one should have been named that originally, it would just be a bigger mess than it would help fix. So, again, maybe I'm rambling, but those are some thoughts on, DNA as a magic bullet or whatever. So, yeah,
absolutely. I do think it kind of falls into a box of the, as you say, the sort of veneer or illusion of certainty, right? In the moment, this is the best that we can do. And we sort of then take that both retroactively and sort of see that as the deficiencies in past things, right? So as you talked about, it's sort of the evolution of even what would qualify as a viable species description was different 50, 20, 100 versus 50 versus 20 versus 10 versus now. And then the same way going forward, I don't doubt that they'll be incredibly different, right? It kind of fits into, to some extent, it's important, as you say, to just recognize the element of uncertainty. The thing I think about when you were going off about looking at these DNA strands, right, and you'll know way more about this than I do, but is the ability for or human DNA containing the DNA of viruses and things, that are just sort of retained into our features and things. And so what does that mean? So theoretically, right, if it's entirely derived down the line, then we could see, okay, maybe here's where it popped in, and then you could see descent down the line. But do we even know that with the element of certainty that we think we do, right? When we're presenting it. When I would present it in that way, you'd say, well, maybe, right? Yeah. I would think. Or
it's the best we know now. And so that's what we go with, nobody's going off of the worst information. Although sometimes you wonder. I mean, I think even the concept of a species has changed a lot over time. And now there's several different species concepts that may take precedence in certain circles or certain
amongst certain types of organisms and things. Yeah.
I would venture to guess that most people listening probably have heard the species concept, that if two species, if they're indeed separate species, they will breed and produce sterile offspring. The mule is the classic example between a horse and a donkey, and you get a mule and they're sterile. And so that means donkeys and horses are separate species because their offspring are infertile. But that doesn't apply to reptiles at all, because you can get all sorts of crazy hybrids between the most basal and the most advanced or evolved or recently evolved. And you can get, fertile offspring from those breeding. So obviously that species concept is outdated and does not apply to reptiles.
Right.
For an example, for a very simplistic example. And there's all sorts of, there's species cloud or, those kind of concepts where, there isn't just this is this right now, but it's moving in this direction, or it interacts with this different element or species group or whatever, ring species. There's all sorts of cool concepts out there. So, yeah.
Yeah, absolutely. I think the other thing to hit on, right, and we're not stuck in sort of the vernacular portion of this, but would be to say that at its most basal, right, a lot of taxonomy, scientific nomenclature and things, the intent is to avoid the problem of common names so that at least we can ensure we're talking about the same thing. Yeah. And to that end, right, even if we're in a space where I'm using the name that was proposed or accepted on date X, and you're using the more recent nomenclature, X plus 10 or whatever it would be, the process of that records what the name was previously for that organism. So if we're both sufficiently informed, that can instead reflect sort of what that should reflect is our willingness to accept the argument that was made in the paper that you're referencing, right? You probably previously were using the nomenclature that I'm still using. And instead it's, that's the conversation we're having, not a fundamental disagreement about what it is that we're even talking about. Yeah. And that's really kind of the whole point of the whole thing, is trying to put labels onto stuff so we can all be talking about the same thing, as opposed to just saying the brown snake, whatever that might mean. That would mean something different to someone in Florida, right? Maybe they think brown water snake, Texas Pelota, versus if you're in Australia, obviously brown snake, there are many brown snakes, and even amongst that, which one are you talking about? So
And there's some erroneously named brown snakes, like a king brown, which is actually a black snake.
Exactly. Yeah.
So I had a fun conversation with Chris and Aspen. We were herping and we found a chuckwalla, and I said, "Oh man, their scientific name is so fitting, Obesus." And they're like, "What are you talking about? It's Sarmalis adder." And I'm like, "Adder? When did that happen?" Like, no, it's Obesus. Like, that stuck in my mind from when I was a kid. And I guess I never revisited Utah reptile taxonomy or whatever, because I hadn't seen the change to Sarmalis atter. I had it in my mind that they were still Obesus. And so I was corrected and acknowledged that correction. And now I refer them as adder, but I still think Obesus is such a much better name.
Yeah. What you need to do is go back to that paper and then see if you actually agree with the change, right?
Yeah,
and
there you go.
You can fight Aspen in Australia and say, absolutely not. It should be Obesus. I read that paper. That didn't make sense.
Yeah. I disagree wholeheartedly with their change. And it probably is something like, well, originally they were given this name. And I mean, we find that with the python we were trying to find out in Australia.
Yeah, absolutely. Absolutely. And I guess the reason I tend to not engage with that conversation quite so much is because some of it then just turns into silliness, right? Yeah. On the one hand, it could reflect, why are they all Gonyosoma, not Rhynchophis? It's, the Gonyosoma name was applied in 1889, and, Rhynchophis, that was 1893 or 1897, this sort of, and well, it's probably not totally accurate. Certainly the conception I have of that point in time, that just seems sort of silly, right? In some sense of like, okay, where were these specimens coming from? How were they being published? The whole going back to the sort of dynamics of it. Oh, you need to distribute X number of copies within a day's distance of your location of publication, and all the stuff that reflects the fact that this system is born out of the 1700s and sort of what technology looked like and information sharing looked like and all those things. That I try not to get wrapped up in that, because some of it's not a question of it's solely almost priority and then uptake, as opposed to whether it makes sense, sort of how inclusive or exclusive it is of intrinsic meaning, right? As you highlight and Oenpelli pythons. Nyctophila python, I recognize that name exists from the mid to early 80s, but translating to be night-living python, and that's just absurd because my intrinsic challenge there would be, okay, which distinguishes it from literally every other python? How?
Yeah, exactly.
As far as I know, right, in appropriate temperature, they're all nocturnal. So I don't. That doesn't make a ton of sense to me.
Help. Yeah.
Yeah.
And Noirin is
just descriptive.
Such a much cooler name and cultural and
exactly right. Right. And so, I recognize the need to have a system, and that's fine. But that's why I tend to, in some ways, it's not as interesting, right? I can poke fun at it or whatever, but I try not to get super tied down into that rabbit warren, because it can be very frustrating to say, okay, recognizing the system of priority for hundreds of years, this is the way that it will be. It's not necessarily based on the overall quality of the description or even the intrinsic meaning and things like that. It's just sort of, we just give deference to what came first that was recognized as being legitimate at the time.
Yeah. Well, and two, some of the names, the species were named after those people turned out to
absolutely
be
same issue,
Nazis or whatever, and you're just like, now we got to call it by this, honor this guy who was a horrible person or whatever, or this person who had no clue what was going on, or, had no interest in reptiles at all, probably was the number one murderer of snakes in their neighborhood or something, but because they were the Duke's son or daughter, they got named a snake after them. Anyway,
and some of them are great, right? Sure, on the other hand, and then some of them are funny, right? And you'll know that I don't remember the first and middle initial. I want to say like AG or something, but like Children's Python is named for a person whose last name was Children.
Yeah.
Right. Yeah. Or children's. I don't remember if it's singular or plural. Yeah. But so it's like people, whereas now if you take them to a show, I'm sure people will think, oh, this means they're appropriate pets for children. That must be why they called them this. And it's not. Right. So, super interesting and convoluted, and invariably, right, as we move forward, almost inexorably, the things of the past will look worse relative to modern conception. That's just sort of the way of progression, progression, whether it be or not. Right.
And I think there is a push to change that system of naming, so you don't just have everything be hosere or, it starts to be more descriptive of either the appearance of the animal, the area it comes from, some kind of cultural reference to that animal and in its importance in the native people, so I agree with that. I like that type of move. I like a descriptive, stellatus or something, the stars, you can picture the spots on the back being the stars in the sky. That kind of thing is kind of a cool thing. Or, like our Noirin, a cultural significance. I just like that name. And you were welcome to use that name because it is the Aboriginal name for the species.
Absolutely.
So, and we'll know what we're talking about if we say Noirin or Noirin, whoever. I don't even know if we're pronouncing it correctly, but their Americanized version of whatever it's supposed to be. But I think that's kind of a cool thing. I guess we won't even touch on pronunciation and how things should be pronounced. We have some fun. We've had some fun discussions in our group of different pronunciations and Willardy versus Willardi. I don't think his name was Willard. I think it was named after a guy named Willard. But anyway, keep calling it Willardy if you'd like, Nipper. we won't poke too much fun of you. But just the idea of, naming, as long as we kind of recognize what they're talking about. If we say Nyctophilopython, or if we say Morelia oenpellensis, we'll know, I know what you're talking about. You're using an older name, but that's okay. Maybe Obesus and Adder might be confusing because, Chris and Aspen had never heard of Obesus. That was not familiar to them at all. And they're, 10, 15 years younger than me or more. So they wouldn't have, been a kid looking at the
absent
field
looking
guides
at
and seeing Obesus. They would see Adder in all the guides they had. So it's understandable and kind of fun. But
I don't know. I thought Aspen had presented himself as having gone through and was studying, be it Old Field Guides, Wikipedia pages, studying all the preprevious names so that he could engage in that conversation with you. I thought that was how he had presented it, but maybe
it could have been. I probably remembered that, but yeah, it's funner just to say, "Oh, these guys are young. They don't remember the good old days when it was Obesus."
Oh my goodness. And certainly in terms of the whole sort of politics of naming, and as you described it in terms of sort of, for the most part, what the naming paradigm looks like now, Herpetological Highlights has had several episodes where they've really kind of talked about that. And certainly they, seems like the view that you espouse really reflects sort of the view they have as well in terms of descriptive, be it of the animal itself and its appearance, have it be helpful in some way. Right.
Right.
And even Dave and Tracy Barker, in terms of the names that they would suggest, were really proponents of that, of trying to promote clarity within both the scientific and particularly for them, the common names of things, right? It should be a lesser Sundus python, not a Timor python. That's sort of an idea.
Yeah. The Perthensis, Antaresia perthensis should probably be Antaresia pilbarensis instead.
Yeah.
They don't come from Perth. They come from the Pilbara. But yeah, I mean, there's always going to be, fretting about naming in that regard. But yeah, I guess this isn't necessarily this discussion. I think back to the idea of presenting their, putting their best foot forward. I do agree that a lot of papers will put forth several different ideas and they will go, they'll kind of choose the one that they think fits best, like you said. And so, they're going off the best that they've received or the teachings or philosophies of the time. It's really hard to go against the grain and try to introduce a new species concept or a new, nomenclature concept or things like that, because you really have to make a very, very strong case to do away with what's been done in the past. And so, or
other people are doing in the moment, right? That's
exactly.
the whole thing, right? And the other part of that, I think that's a great point. I hate to jump in on you, but saying like they need to acknowledge, a paper that's making to be taken as a rational argument. It needs to present where there are clear alternatives, right? It's not only there's actually self-interest in saying, well, I'm choosing A. I recognize that it could be A, B, or C, but I'm choosing A. Otherwise, they'd be exposing themselves. If the paper just said it's A, that's it. Yeah. That's
don't worry about anything else.
To being, very vulnerable to being attacked both, in the peer review process and upon publication, in the sense of saying, well, actually, it could be A, B, or C, and you didn't even acknowledge that B or C could exist. So we're going to impute that you didn't even perceive that, rather than saying, okay, you recognized it could be A, B, or C, and you chose A. So that's actually, linguistically, it's important for them to present those choices and be honest about then making a choice. Otherwise, it looks like you just didn't even realize that it could be that choice.
Right. Yeah. And I think there is some sense of, how to put this, when you say things have to be kind of on their own evolutionary trajectory, and that's really tricky to say what, constitutes an evolutionary trajectory. And does, we kind of discussed a little bit before the show about, the, Australia, New Guinea connection, when, the ice age occurs and the sea levels lower and the land appears, and now all the different carpet forms in that Lake Carpentaria area, now that's a lake instead of a gulf, now they can move around and interact and interbreed and things. Now that happened several million years ago. What we have now has been isolated for several million years. For example, a laboratory mouse breeding center. Okay, how does this fit in? Listen and let's see if this helps. But they have this laboratory, research center that breeds rodents for research, right? And they have specific strains of mice that they breed and they're actually inbred strains. So they breed siblings.
Isolating certain characteristics or traits.
Exactly.
Right?
Yeah. And so these have been bred for generations, inbred for generations. And so say company A will have this line of mouse, we'll call it, strain B. And strain B, and the company gets bigger and bigger, and they have more demand for strain B. So they have, now we need to ship to, all around the country. So we're going to have a facility in California and a facility in New York. So they split strain B into two, and they put, one colony in California, one colony in New York, and they're breeding this strain. They're not introducing any other strains or any other mice. They're just breeding strain B true. And they do this for, 5 to 10 years. And then they compare strain Bs between these two facilities, and they find 20,000 different changes in the genetic code between the two facilities. Is strain B the same strain B that in California as it is in New York? Not so much anymore. For an example of how this affects in the real world is, we have, an infection, forgive me if you don't agree with research in, animals, but we have viral models of, in mice, and we infect mice with viruses. And so sometimes we would get a different outcome, and we'd say, okay, we've infected this strain with influenza before, and, it's lethal in some cases, and then in other cases it's not lethal. Well, we get to digging and we find out, it's lethal when we infect mice from New York, but it's not lethal if you infect mice from California. And so it changes your whole research. And so, kind of the conclusion was, don't order mice from, the California facility or whatever, get them from the New York facility, because those are the ones that are, whatever their genetic code makes them susceptible to the virus that we're using or whatever. So, yes, those changes do have an effect. Are they still the same strain? Well, yeah, they're obviously still mice. They're obviously,
still mice from that same
thing.
Yeah. Exactly.
Are these changes enough to say, no, they're not the same strain? Should we call them substrains or,
right?
sub-strains? What, how do we differentiate those two? So we differentiated by facility. Now, could the same be said of, these populations that have been isolated for 5 million years? They're going to change quite a bit between Darwin and Papua New Guinea and Cape
right.
York, and they're going to change to match the environment. They're going to be isolated to some extent because there's no carpets swimming in the ocean, and there's no carpets kind of in that Lawn Hill area at the base of, the lake or Gulf of Carpentaria. And so there isn't currently any intermixing going on. So they've been isolated for 5 million years. I guarantee there's plenty of different changes that one population has that the others don't, and they're having their own unique changes. But what is enough change to constitute a difference? And what is enough time to say they're on their own evolutionary trajectory? Because obviously all the individuals now are not going to be intermixing with the other populations. And
right.
So they are on their own personal evolutionary trajectory. Now, as a group, what does that mean? And how long does it take to have sufficient number of changes to say we are different now, we are unique? And when we join back up in 5-10 million years or whatever, are we going to be able to mix and mingle again and form some new, that, I guess that's maybe the species cloud concept, is they're ever changing and ever evolving. And Ben always liked to throw this one. Ben Morrill used to throw this one in the mix when he'd say, as soon as you take it out of the wild, you are taking it out of that evolutionary trajectory. You're putting it on a new evolutionary trajectory. So technically it stops being that species from where you got it from, because you're choosing who it breeds to, and you're right. choosing what, it experiences. And so you're changing it in certain ways to mean that it may not fit in with that species that it originally came from. So, that kind of maybe illustrates the point of how challenging this is to put a label on something that's constantly moving. And so we do our best. We try to say, okay, this level of change is fine, but if you go more than this, then you're this, or, it would be nice to have that. I don't think we do have that, because I think in maybe certain areas you do that they care about, percentage of genetic change or whatever, but I don't know. It's a complicated topic. It's very a complicated thing. And so you do just have to form the best case that you can and say, look, these guys are separated from these guys by this biogeographic barrier that tends to have speciation on either side. So we would say that probably constitutes their own evolutionary trajectory. Now, maybe they can cross once in a great many years, and maybe this one gets in and interbreeds. Or,
right.
You've got the case of the diamond pythons and the coastal carpets, and they have an intergrade zone because they're different things, and they interbreed and make a third thing. So how that you could use that to say, well, they're the same species because they interbreed and it's fine. Or you can say, well, they interbreed and form a third thing, so
right.
they're a different species. This is a hybrid, not a intergrade or whatever. So it becomes complex.
Yeah, absolutely. And what is there is not the same thing as if you just tossed a diamond with northern coastal, right?
Mm-hmm.
It won't look like that same thing. It's not that simple, right? It's so many iterations upon one another. And this is probably self-evident, but right, not all biogeographical boundaries, even of the same type, are the same in the sense, as you were talking about the sort of northern Australian to southern New Guinea, I'm thinking at the same time in that same general area, isn't Biak or Biak, right, is the term continental island, right? So it's just that the gulf there between northern New Guinea and Biak or Biak is it's super deep water, super deep current, quick currents and all these things.
The island of Biak was never a part of New Guinea.
Exactly right.
Separated by deep water. Yeah.
Yeah, so which is just super interesting, right? So it's not the same thing as, something in the Torres Straits relative to either Northern Australia or Southern New Guinea. Mm-hmm. So just the level of complexity is amazing, right? Yeah. And in terms of if you're talking about speciation on Biak versus in the Torres Strait, how would you apply that same standard? Or if we're talking about genetic isolation, those inherently will be different. And your argument will be more persuasive if you're talking about potential speciation of something that seems very similar in Northern New Guinea and in Biak than, something in Southern New Guinea and the Torres Strait.
Mm-hmm. Yeah. And I mean, you could even... for example, just thinking about kind of the Biak versus the other northern New Guinea, Biak wasn't given a species of green tree, right? Even though there's probably some evidence that they probably should be a different subspecies at least, or whatever you want to call them, because they are fairly isolated and they've been, they've diverged, they've been divergent for quite a while, and so There are definitely some scientists that say, no, there should absolutely be a subspecies of green python from Biak. But the last paper, for whatever reason, didn't make that happen. So, there's always things that are either missed or undersampled or whatever, for whatever reason, or the region that they looked at was not the region of change for that particular isolate or whatever. I think about the pygmy banded pythons with the latest Antaresia paper. They're not even mentioned or included. I think there might be a picture of one in the paper, but they're not involved in the sequencing or the DNA differences.
Right. Some of this comes down to sample, right? The samples that are available, literally, did they have samples of this form to be included in the paper or not? Those practical considerations shape this more than, again, going back to the old bugaboo, right? Of, this is, science says, and it's like, no, all of this is occurring within the context in which it takes place, right? There are practical constraints on all of this. And
if you ask any herper that knows much about pygmy banded pythons, you would say, no, that's a different species. That's not a Stimson's, children's now, as the argument goes, or a spotted. They're not neither, and they overlap with both spotteds and Stimsoni or childreni. Frankly, I think those eastern Stimson's probably are children but anyway, they're something different. They're smaller, their head structure is different, their scalation, their counts, their measurements are different. So why did a paper looking at the genus Antaresia miss that? You know what I mean? Like how, and I know they were aware of them because I believe there's a picture in the paper, and I know for a fact that Peter Birch had talked to them about this putative species or whatever, and they just basically ignored it. So who knows what the reasoning or what how that occurred, but we still don't have any scientific evaluation of that, putative species or whatever you want to call it. So that's kind
right.
of
And part of the thing that jumped to mind, right, as we're having that conversation is like, well, fundamentally, right, it would suggest that they are indeed different. The fact that they occur conspecifically with both those other forms, but exist as themselves in that area. Yeah. Although then, kind of gnawing at the back of my mind is the thought process that we've seen with California king snakes, with Alterna, right, of saying like it wasn't until they were born in the same litter and it's become clear that there are multiple forms within a given population, and there is some regional variance to prefer selective pressure to prefer the striped ones versus the banded ones. But in both those contexts, we're talking about things that were described as being distinct, and actually they're just the multiplicity of forms that are within that same grouping.
Sure. Yeah. I mean, obviously, it's a very complex issue. That's why it would have been nice if they would have looked at them genetically, or
sure,
or done
that's
scale
that'd be
counts
useful,
or measurements, right? Or whatever. Yeah, that would have been very useful to have in that paper. So that's
or hemipenal structure. They ever loved.
Yeah. Dirty taxonomists, always looking at the hemipenes.
Although that's so interesting, right, in terms of like the reason that those diversity. Different structures exist, why that diversity exists. And that actually maybe as a speciating factor is one of those things that was looked at hundreds of years ago, and actually maybe should be looked at more in our present construct, right? As opposed to just deferring to DNA, because that literally we are talking about something where you need the key that fits the lock, so to speak. And that actually is a limiter.
Right. Yeah, that is an interesting point. Oh, yeah. If we could standardize this, that would be nice. I mean, obviously there's some things that precluded the ability to standardize these kind of things or to say, but at the same time, I mean, it would be nice if, for example, there's a new paper, Vertebrate Zoology from 2024. It's called Vickers in the Desert, Substrate Specialization and Paleoerosion Underpin Cryptic Speciation. I love the term cryptic speciation in an Australian arid zone lizard lineage. So basically they're talking about substrate specialization. This is referring to the helmeted gecko, the Diplodactylus galeatus, and they've found that actually consists of 3 different species that have specific preferences for 3 different substrates. So
interesting.
And, a fairly not a huge ranging gecko. It's not covering all of Australia. It's kind of Northern Territory, northern South Australia, kind of a little circle in there. But they've found that there's enough in their minds information to warrant the splitting of that into 3 different species. And this is McDonald, Fenner, Tarcola, and Paul Oliver, who Peter J. McDonald, he's done quite a bit of this kind of thing. So, but I do like including that kind of thing of their interaction in the natural environment.
Absolutely.
I don't know how you get around that and not have that be included. Now, your point about the California king snakes, I think definitely, that could play into this, and you can have the same species have different looks in different areas with different types of soil. But, when do they draw the line into saying, I'm only going to live in this type of soil? I'm not going to range on the other soils. And so when do you stop being, a variable species?
A singular species with a ton of variation into these hyper specific, right? Anytime we're talking cryptic speciation, that generally means if someone, even an informed consumer, so to speak, were to be looking at the 3 different species of formerly Galeatus, right? cryptic speciation suggests that they look more or less the same, right? You need those extra, the information on the externalities and how they interact with it for it to be, okay. Now I can appreciate this difference. Yeah.
Now there are some kind of general rules of the differences in pattern to some extent. Like some have, more white spots. Some have, a certain species have more white spots. Some have more portholes along the side rather than, diffuse spots. And then one's kind of intermediate to that. They have this portholes or spots. And so it's just one line of, that kind of idea. So there are some hints of differences. And, that was similar to the Wheeleri. They were both the Nephurus, the knobtails, the banded rough knobtails. The common names are almost harder than the science. You had Wheeleri and Cinctus, and they were both subspecies, but then the paper came out showing sufficient argument to say that these are full species and they should be recognized as such. And so now we've got Wheeleri and Cinctus. So, which is, they see those differences. They see the pattern differences, the structural differences. One had a more blunt head or a shallower head or something, and you can show that consistent measurement between the two groups. And then eventually that pans out and with the genetic analysis into, no, this is really, something that differentiates the two, because we also see a huge divergence in the genetic code as well. So kind of interesting, but somebody could come along and say, you're crazy. This is just this is in
your imagination.
Yeah, exactly. There's just slight differences in their patterning because they're on different soil types. And, you're saying it's because they have to live on those soil types. And, so there could be an argument the other way. Now the genetics, they might, kind of overlap to some extent, and in some of the morphology they overlap, but I guess you could make the case, no, there's definitely some divergence there, and they make a reasonable case. Now maybe if I was a Galeatus keeper and I studied them and found them all throughout Australia, or their range in Australia, then maybe I'd have a more informed thing, similar to what I have with the Antaresia, where I'm like, nah, this is nonsense. There's definitely a difference between Stimsoni and Childerni, especially the Western stems. But anyway.
Yeah. Well, and I mean, so the thing that was jumping to my mind as you're talking through that is our own experience with the Shai in the Northern Territory, right? So prior to, and you'll know this history way better than I will, but if you go up the chain far enough in terms of sort of the history of the naming conventions of these things, all of the Asper/Amii animals were Asper, right? Until some point in the 80s, late 70s, 80s, something like that, right? So Amii as a distinct entity was recognized at that point. The part that was jumping in my mind is that animal was so much more, not having seen an Asper in the wild, it was certainly reminiscent of all the things that I consider to be Asper characteristics rather than an Amii. So the idea that, when I'm reading the reptiles from 95, Casey, it's got the Amii on the cover and all this stuff, the idea that, if you went to Kakadu and found one of these animals, would it be an Asper? I think that article, I think everything is Asper. Maybe it even frames, it doesn't recognize Amii, possibly. But the question would be, well, which would this be? And, from that perspective, it's like, no, that was an Asper type, not an Amii. You wouldn't, having interacted with it, there's no confusion that that's an Amii.
Yeah. That's always and they say, well, this one's in the red center, so it's orange, and this one's up here on the rock escarpment, so it's a little darker. They just kind of. But then when you get more information, you get a better picture of the range, or of the variability within the range, and you see, okay, I'm starting to see patterns here. Well, there's this huge gap where you don't find any, or nobody's found any. So does that mean they're not there, or they're not any roads there, And so you kind of start to build the picture and say, okay, I'm starting to see a pattern here, and then you see, obviously Amii are very different than Asper or Shai, and there's huge gap in the distribution above, AMI between AMI and Shei and Shei and AMI and Asper.
Right.
And so then you say, oh, obviously, how could you think these were all the same species, because you didn't have the information that we have today, all the range and genetic analysis or measurements of the different species. And so, it's a little harder to see from a limited information.
Yeah.
But you're right. When you're looking at old papers, you have to kind of look and see in the materials and methods where they were collected from, because some of those Asper may have been AMI and not Asper.
Right.
That's what.
And that could cause some of the confusion of saying, there's all this variability. And in reality,
exactly.
That's what it's hiding. I mean, I guess the part that was so visceral for me was just saying kind of the very light, framing around Shea had been, it's AMI with the, the toe bands, or whatever. And it's like, then we're looking at this thing and I wouldn't describe it as that at all. That's not correct at all. And that could reflect sort of just the very casual nature of my exposure to it, right? You having written the book, undoubtedly it was less of a surprise to you, but that was just sort of how it had been colloquially framed in the early 2000s. They're calling this thing different, and it's an AMI with toe bands. No.
No, not even. Yeah. Right. Yeah. And I think too, I mean, we were talking on the trip about the glauerdi and how you can find glauerdi in Kakadu, and they're so disjunct from any other glauerdi populations. And how is this even a glauerdi? They look different, they act different, they climb on trees, or that kind of thing.
Yeah.
And then you're thinking, man, has nobody? But obviously somebody's been thinking about it, and somebody's been working on this, and these things kind of move slow sometimes. And so, having the patience and there's always whispers like, oh, so-and-so's working on this, and they'll have a paper out, and it's like 10 years later, someone's working on this. They'll have a paper. It's not
working diligently.
Exactly. Yeah. And I mean, it's hard because, the funding's low and nobody's paying people to go out and collect samples, or, you might not have enough.
Even is it permissible?
Yeah.
Right.
Yeah, exactly. So you're reliant on collections, what they have at the time, and, those kind of things. So there's a lot that goes into this, and it's a very complex thing. So it's hard to judge the taxonomists too harshly, because I do think that they're doing the best they can with the current knowledge that we have. And so, this isn't to say you have to agree with everything that comes out. You can have a difference of opinion, and, you can inform yourself, learn about these concepts and say, no, I think it probably fits better in this arena. And maybe that drums up some, you can publish your own paper. There's nothing against, citizen science. Might be best to tag team with an actual scientist that shares your views, or you can kind of convert over to your side, I guess. But there's nothing to say you can't do that and set the record straight in some cases,
Just distribute it. You need to print it and distribute it within a certain number of copies within a certain distance of your house, and you too are abiding the rules of the 1700s.
Exactly. And I guarantee there was somebody out there saying, AMI aren't this northern, central Australian Asper is not an Asper. It's an AMI. It's something different. I think we should name it after this explorer's daughter. So there's always somebody out there seeing these differences and making those. And I think that's an important part of this too, as we go out and find weird things and find animals that don't fit a certain box, like, oh, this looks like a weird glauerdi. Well, maybe it isn't a glauerdi. Maybe it's a weird something else, So that's the idea, is keep
trying to bring an open mind. Yeah,
exactly.
An open mind both to the possibility and innovation, right?
Mm-hmm.
And I mean, that's the part of feeling like I don't have appropriate expertise in this, is just the constant innovation. I know that there's stuff that I'm, I know how much I don't know, as you highlighted earlier.
Yeah. And it is hard to just kind of jump into the, it would take quite a bit to, get to the point where you're going to be able to know all the rules and follow the thought of the day in regards to taxonomy, but
and have the technology and capacity and all these things.
Exactly. Sure. But these days, I mean, anybody can hire a lab to sequence a genome. You might even be able to. It's hard. You might not be able to get into some of the collections unless you have credentials and things like that. So there are kind of some gatekeeping, barriers in academics, which sometimes can be good and sometimes can not be so great, so, but there are ways to get access. And if you're serious enough and you have good enough concepts, there's a way to do it, there's probably somebody sympathetic that does have the credentials that can help you out and get you there. So I would say anybody going down that path, keep going, fight the good fight and make your case heard, because you may have good information that just isn't well known in the scientific community. So I think we assume if they're a PhD, they should know, much more about this than I do, but that's not necessarily always the case. Sometimes that passion of, a hobby or, that we just have this desire to know as much as we can, and we're traveling to different countries to see these things in the wild, and we might not see tens or hundreds of them, but we might see 5 or 6, and who knows, maybe that observation could help put a something in the right direction in regards to that animal. So again, I mean, the pygmy banded python is a great example of that. Right? I think Casey Lazick found one back in the late 80s, and Peter Krause had known about him as well. And so back in the 80s, but there were kind of hints and whispers about this weird pygmy Stimsons, I think they called it. And they started breeding them and had them in captivity. And now they're commonly kept and bred. They're in Europe and even in the US now. So I just talked to somebody, a friend on Facebook that brought some in from Europe. I'm like, oh, that was easy. I didn't think they'd be able to do it. I thought they weren't letting stuff out of Europe that, originated from Australia, but she got them in. So kudos to her. But yeah, so there's all sorts of cool things that just out there to be discovered. So I don't know. I hope that was a reasonable discussion and we didn't.
Some interesting points there. Whether we kept the thread or not, I don't know. But nevertheless,
that's always the hard thing, is keeping us on. So hopefully we got to some of the points you wanted us to make, Mark, or we made a reasonable discussion out of this that you've enjoyed, and our other listeners as well. But that's I guess where we'll leave it for tonight. And maybe if you have some other thoughts on the topic, we can keep this up at another time. But I think for tonight, my brain is not functioning well enough to go further. Yeah. Well, any cool things out there that you've seen in herpetoculture or herpetology in general?
I guess the thing that's really jumped out at me is just the incredible content of wild herps on Instagram, right? All these different accounts that I wasn't aware of, that I just stumbled onto one. Somebody who, a local in South Vietnam, who's operating tours to both South and North Vietnam. And as you can imagine, as I'm flicking through the different pictures, the thing that jumps out is, I see that's a frenatum. Have you seen rhinos? And they're on there twice. So I'm going to assume that he's gone and found him at least twice. So that's someone that I'm interested in talking to. So, yeah.
Very nice.
That's super interesting. And just as one specific example too, I mean, there's so much content out there. It's sort of overwhelming. And the key is just to keep the right perspective of, taking it as additive and fulfilling and things, not, we're sitting here in the snow, or whatever. But no, all good.
Yeah, a lot of armchair herping and exploring to be done, right? I found the same thing. I got on to Flickr. What a great resource Flickr can be, in a lot of ways. It's just a photo hosting site, but people put a lot of photos on there and of wild herps. There's just some phenomenal photographers on there, and you see some amazing herp photos. Another cool thing was Gavin, when Gavin Bedford put out a request for photos of Kakadu glowerdi, and they had like, a bunch of responses with photos of Kakadu glowerdi. And I'm like, oh my goodness, that's amazing. So, I would love to keep seeing photos of Kakadu glowerdi, and
absolutely.
It's interesting how much you miss, what relatively little you're able to see just because of the immensity of the volume of content. You get on to those social apps and they dictate what you see, and their algorithm tells you what you want to see and understand. But yeah, it's cool to kind of do your own searches and find targeted species or whatever. So Flickr is a fun way to go about that. And a lot of times they'll say where the photo was taken or the general area or whatever. So you can kind of say, oh, that's what they look like in that area.
Right.
Cool. Yeah. So I've obviously been on a little bit of a glowerdi kick after the whole
I can imagine
experience. Oh, I did post a YouTube video on my search for glowerdi. So it's a 5-8 minute video or something, and it kind of
I'll have to check it out.
Shows where I went and kind of my thought process,
conditions, and
hiking out and things like that. And then it ends with the really close footage that I got in
nice.
Up in Kununurra. So cool. Fun stuff. I'm still just geeked out. I'll still watch the footage and just get excited about it, So
yeah.
And I've been putting up, going through my pictures and putting up photos of,
I know that Michelai picture that you just put up was really nice.
Turned out really good. I'm pretty really happy with some of the photos I got. So I'll just be kind of putting those up every day or two. Whenever I get the whim to, oh wait, I need to post a picture of this species that I saw, or this one. So yeah, I've got plenty of fun pictures. That Scalaris picture turned out really cool too. I love their poses. He's like, doing the Spider-Man thing on the tree. And I've got a few that are, even one where he's like holding onto the tree this way, but then his head's turned all the way around looking back at us. It's pretty cool.
That's absolutely cool.
I
need to do the same. Yeah.
There's just so much. I took way too many pictures. And then when I get home, I'm like, man, why didn't I get more pictures or video or whatever? I didn't get enough.
It's never enough.
Yep. Right. Oh, well, I'm still dreaming about that trip. That was a trip of a lifetime. It was fantastic. But now I want to go back. I have even a better feeling for what I need to do and where I need to go to get that accomplished. So maybe another trip to Darwin area is in order at some point.
I think that's sort of the way it goes, right? Are you ever really, and maybe foreshadowing, maybe that'll be a topic for a future show of saying, can you truly check off a place? Can you see all the things ostensibly that are there? Or do you, we've hit on that before, but there's a specific idea, associated with that. So.
Yeah. A little teaser for what's coming up soon in our Reptile Fight Club future. So stay tuned. Keep listening to us. Hopefully we haven't, I was looking back on kind of the episodes and when they came out, and I'm like, oh man, some months we just weren't very good at recording many episodes. Some months we only have one, and others we have one a week. And so we've kind of been, hit or miss, I guess, a little bit. So I apologize for that, but hey, it's free. You get what you get. We're taking time out of our busy schedules to do this. So But if you want to see more content, make more suggestions. Come on and fight with us. Yeah. But
absolutely.
Anyway, after that, being mean, I'll say thanks for listening, and we'll catch you again next time for Reptile Fight Club.